Okay? CD4 cells. This is all of this sort of stuff, which is, yeah, I wanted to fit it into one page, so it just goes a bit dumb at the bottom. Right, so CD4 cells, what do I want to talk about? Talk about first versus second antigen presenting cell. Okay, so I've talked about, what's the first antigen presenting cell? Okay, what's the second antigen presenting cell? Is it anything? For what? For what? For CD4 or CD8? Okay, almost right. Okay, remember, first antigen presenting cell is a dendritic cell. The second one, so when I say what's the second antigen presenting cell, the answer is, you should be asking me, for what, the CD8 or the CD4? For the CD8, it's everything. Any cell can be an antigen presenting cell, apart from like red blood cells, so they don't have anything, right? So for a CD8 T cell, anything can be the second antigen presenting cell. For the CD4, you have certain specific ones that you need to know. So this whole Th1, Th2, Th17, Treg, all of that stuff. So Th1 macrophage is the second antigen presenting cell. Th2 is B cells, is the second antigen presenting cell. The Th17 is neutrophils. And Treg is a complete separate class on their own. You don't need to worry about them. Treg stands for T regulatory, so it's the regulation of everything else.
What's the Th2?
Th2 is B cells. Okay? So you understand the difference between the first and second, yeah? The first antigen presenting cell causes, I don't know if this answers well. Okay. First antigen presenting cell causes the mature naive T cell, the Th0 cell, to differentiate into the Th1, Th2, blah, blah, blah. The second antigen presenting cell basically fully activates the T cell. What I mean by fully activate is it now has the ability to activate whatever fully activated it. Okay? That means that whatever the second antigen presenting cell is, basically you have a second, this is just for CD4 T cells I'm talking about now. The second antigen presenting cell in the life of a CD4 T cell, the purpose of the second antigen presenting cell is something that basically wants help from that CD4 cell. So that second antigen presenting cell is something that wants to be activated. In order for it to become activated, it has to first activate fully the T cell, okay? And then the T cell has the ability to activate the second antigen presenting cell. Is that confusing or is that okay? Yeah. Yeah. Okay. Does that make sense to everyone or is that a bit too confusing?
Can you go through what you said about T1, whatever, T17?
Oh, okay. T1 macrophages, T2 B cells, T17 neutrophils. Macrophages.
Yeah.
T17 neutrophils.
Wait, you said just T1, T2 as well? Do we
T-
just know this? I'm not going to write stuff.
Yeah, you do.
Okay, so
Yeah.
T2.
T2 B cells.
B cells, okay.
T17 neutrophils.
Okay, is that it?
Yeah. Okay? And then Treg, don't worry. Treg regulation.
Don't worry.
Yeah, but you need to know it exists. I haven't just quoted it for fun. Yeah. Yeah, I'm going to say the activation thing. I'm going to draw a little picture if that helps. Okay, guys, look at this. Okay, guys, look at this. Shh. Guys, shut up. Okay, this is a Th0, okay? This is a dendritic cell, so it's the first antigen-presenting cell, okay? When these two interact, this goes on to become a TH1, TH2, TH17, one of those. Okay? Let's say when these two interact, it becomes a T1. Okay? Now, this is, think about it as being semi-activated. So this is not activated at all, this is semi-activated, and now a macrophage, which is the second APC for a T1, yeah, macrophage is the second antigen-presenting cell for this T1, comes along, is wanting to be activated for any reason. For whatever reason, it wants to get activated. So it pops along, interacts with this T1. This T1 now becomes fully activated, so to speak. What I mean by fully activated is that this T1 now releases cytokines to activate this. So things like interferon, gamma, stuff like that. TNF alpha, whatever. Okay? Does that make sense? That's what I was trying to say. Now, this macrophage is fully activated. So this goes off, this is activated. And what two things did I say are activated macrophages? So you have giant cells and epithelioid. And these are instrumental in granuloma formation, which is one of the slides in your lecture, in two of your lectures, in Andy's lecture and in Cam's lecture. Okay, so an activated macrophage, you can call it an activated macrophage. Yeah, it's probably easier if you remember it like that. But just remember, if you read it somewhere else, it could be called a giant cell or an epithelioid cell. Does that make sense? Does that whole process make sense? It's the sort of the interaction between these things. They communicate with each other to try and activate each other. That's what immunology is all about. That's what this whole flowchart's all about. Okay? So that is CD4 T cells at a glance.
Sorry, could you go back to the activated macrophage? What does it say at the top? I can't.
Okay, so there are other names for an activated macrophage. The giant cell, epithelioid cell. They're two different types. Okay? One thing I want to say about apoptosis, it might come up as a microanatomy thing. How would you see that something's undergoing apoptosis? Nucleoblemic. Yeah, look at the nucleus if it's got these little ball things in it. You know it's apoptoting.
If it's got what's in it?
Little ball things. Like, it just looks a bit screwed up. And look at, yeah. So there's a picture in Dan's lecture which has got a beautiful picture of that done.
Sorry.
Yeah.
When does it become a CD4 or CD8? That's in the, that's in thymus.
In the thymus. As soon as it comes out the thymus, it knows if it's a CD4 or CD8. Okay? So, yeah. T0 is just, it's not being
That's
activated.
already a CD4.
Yeah, so T0 can be either a CD4 or CD8. I'm just talking about the CD4 side.
But in this case,
It's a CD4. Okay? Okay, so that's, as I said, it's CD4s at a glance. Priming. Okay. When this interaction occurs, okay, certain things that are really important happen. What do you think happens when this happens? One of the things is this, okay? So note this down. When a dendritic cell interacts with a T0 cell, TH0, you get 1, 2, 3 things happening. You get 3 things that happen, okay? Number 1, you get differentiation. So it becomes a T1, it becomes a T2, whatever. Okay? Get differentiation, number 1. Number 2, you get clonal expansion. What is that? What cytokine mediates that? IL-2. That's one of the ones you need to remember. Okay? So you get this, you get IL-2 causing clonal expansion, and you get migration. I talked about migration. Where is this happening, the dendritic cell with a T0? In the lymph node. Okay? It's happening in the lymph node because I talked about acute inflammation and the drainage into lymph node. Okay? But what is the point of having this activated T cell in the lymph node? The answer is there's no fucking point whatsoever, right? You want that activated T cell to now multiply like crazy, so there's billions of them, and then get into the bloodstream, circulate around the body, and kill any organisms that it finds. So those three things are really important. Firstly, clonal expansion, then differentiation, then clonal expansion, and then migration, not only to the blood, but to the site of injury. How does it migrate there? Chemotaxis. Do you see how all of these things come together? That's what lectures fail to do. You fail to get the whole picture overall. Okay? You get migration to the site of injury by chemotaxis. Let's go back to my picture. Okay. Now, I've talked about all of this, so you might wonder, how does a T0 know how to differentiate into TH1, TH2, or TH17? Does anyone know? Different cytokines. Okay? They're usually produced by the dendritic cell, but other cells can help out, like mast cells help out in activation. So what happens is, when this dendritic cell was in the site of infection, okay, it recognizes the pathogen, you get binding, it engulfs it, so it phagocytoses it, it processes everything, and it expresses it on an HLA2 molecule, because this is CD4 we're talking about, yeah, on an HLA2 molecule on its cell surface membrane. Yes? Okay. Whilst it's doing that, it's quite clever in quite a lot of different ways that you don't need to know about. Basically, what happens is it expresses other molecules, co-stimulatory molecules, on its cell surface. And what that does is that will promote which way this differentiates. Does that make sense? Only the first antigen-presenting cell, i.e. the dendritic cell, produces these co-stimulation, and that will cause this T0 to differentiate. That's what differs the dendritic cell from the other antigen-presenting cells. Does that make sense to you all? So based on what co-stimulation is being expressed, yeah, like cytokines, for example, what cytokines are being released, okay, this will differentiate down one of those three pathways. This is where you need to know the cytokines and which ones stimulate what. Okay? So, okay, let's start with T1. So, for T0 to become T1, the dendritic cell produces two main things, IL-12 and IL-23. Let me double check on that. I'm pretty sure I'm right. What does IL stand for? Does anyone know if I'm right?
Yes,
Or am I just making it up? Yeah, okay, I'm right. Yeah, I am right. Super. Okay, guys, I am right. So, IL-12 and IL-23. So what happens is dendritic cell will produce those two chemicals, and that will cause T0 to become T1. Simple. Yes? Hopefully. How does it do that? What do cytokines do? Basically, a cytokine will bind to a cytokine receptor, and that will induce gene activation within this cell, within the T0. It will cause gene activation. That's why he's given you a slide, and it says T-bet on the side. Okay? T-bet. What that means is this cytokine binds to this, then you have this, G protein coupled reaction happening, got a secondary messenger system, and it goes into the nucleus, and it activates this T-bet gene, and the T-bet causes, the gene activation causes T0 to become T1. That's what he's trying to say to you from that diagram. I don't think you need to know T-bet or FOXGT and all of those. Okay? They're just there because he wants you to realize that that's what's happening. As long as you understand that cytokines cause gene activation, that's plenty. Does that make sense to everyone? You okay, all of you up there? Okay? I'm not saying you guys are stupid or something. Okay, good. So, therefore, learn these two. Yeah, for exams, for EMQs. Learn those two. Next.
Do we need to learn that there's cytokines that bind to the cytokine receptors and then that causes gene activation?
Yes. 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 Very much so. Do you know why you need to know? Because what they can do is they're fucking tricky bastards. What they'll do, the examiners, they'll mix up a bit of immunology with a little bit of gene activation. So they'll have a question like an SAQ. This is like one of their ideal questions. They'll have something like, in immunology, this binds to this and it causes gene activation to occur. Describe one mechanism by which the binding of a ligand to a receptor causes gene activation. So then you talk about, you guys have done CAMP and all of that stuff. Yes, cyclic AMP. Yeah, not CAMP.
Just one more thing.
Yeah.
When we say that binds to cytokine receptor causes gene activation, do we only know it causes gene activation? Do we need to know how the gene activation happens?
No. What they can do is basically on what you've been taught in your DNA lectures, they expect you to have a general idea of how that happens. Yeah? So you get transcription factors and all of that bullshit. So they expect you to know. That's why they're not going to repeat it here. So you should know this general principle. You don't need to know how each one does it. But you understand how they can link things up. That's the most important part. That's what they like to do in FunMed, by the way. Okay? They like to link little things up because then they can test you on everything. Okay? And that's what they'll do at the end of your exams. They're really, really annoying that way. That's what PBLs are. Yeah. What you do is they give you 3 PBLs, brand new ones. Okay? One's usually FunMed, one on one of the other systems, and one's an integrated one. Integrated meaning it's everything plus ethics. Okay? And you're left on your own, no tutor sessions, you're not allowed to ask any teachers for help, technically. Okay? You have to work on those by yourself, and they can ask you a 20-mark SAQ on each one of those. So it's half the paper, half your paper A1 or 2, whatever the SAQ paper is seeing PBLs. Okay.
So do you have to do that in the exam? They give it to you in the exam?
No.
You go away.
They give you it like 2 weeks before, 1 week before, and you go over it yourself, and then they'll ask you anything about it. That's where they integrate everything together. Okay, but if it's a FunMed PBL, FunMed Scene PBL, they can only ask you stuff from FunMed. But they'll ask you everything from FunMed. Because they're 20 marks, they're not just going to have like bullshit questions on one little topic.
Yeah.
They'll integrate everything. Okay. Okay, right, you see this, IL-12 and IL-23 and all that stuff that causes T0 to go into T1, T2, whatever. That's what I've described as signal 3 here. Okay? So if you're looking at this and you don't know what signal 3 is, that's what I'm talking about. I've just tried to simplify a little bit. Right? Okay, so that's T0 to T1. Now let's talk about T2. The most important one here is IL-4. Okay, IL-4 is produced by dendritic cells, but more importantly, it's made by mast cells. And what happens is, okay, don't quote me on this, but generally, if you get the general picture, the dendritic cell will interact with a mast cell, yeah, and it will produce IL-4 together, both of them. So you have mast cells here as well, and they produce IL-4. And the reason that mast cells are important is because T2, I said, is for the B cell response. B cells produce antibodies. Antibodies fight things that mast cells are activated by. Okay? Does that make sense? So mast cells interact with eosinophils and helminths. Yeah? I've said that, I think, last week. Mast cells interact with eosinophils and helminths, and they get activated. Helminths are best counted by IgE, right? Therefore, you need IL-4 to convert into T2 response to activate a B cell. Does that make sense? all you need to learn for the exam is IL-4 activates, IL-4 converts TH0 to T2, right? And that activates B cell. Understand?
Yeah.
And just for your information, the gene is called GATA3, G-A-T-A3. Okay. What did I say Th17 is for? Neutrophils. Okay. This one is more complicated, so I don't think you need to know it. But this is activated by TGF. What does TGF stand for? Tumor growth factor. Tumor growth factor beta. So this is by the dendritic cell. Okay. TGF beta by IL-6 and IL-21. Okay. I don't think you have to remember those ones. Okay, that's fine. So don't worry about that. That does Th17. Don't worry, it's all good. Right? And Treg has its own one. Does everyone understand that? Okay? Good, 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 good. If you don't, I've summarized it here for you. Okay. That isn't a mistake, that is actually right. IFN gamma does do it as well, but this is more complicated than you need to know it. That's why it's a degree level thing. Okay, so I said, okay, so we start again on this side now. You have the CD4 T cell, it comes down, it matures, it comes out of the thymus, it's a mature naive Th0 cell. Then you get a dendritic cell, which is phagocytosed something. Yeah? Right, so it's phagocytosed something, expressed it on HLA2. That's the important thing, CD4, HLA2. CD4 recognizes the HLA2 bracket. Okay? You get signal 1, signal 2, blah, blah, blah. It becomes experienced, it becomes primed. And now, with the co-stimulation, which I've called signal 3, it goes down 4 pathways. Worry about Th1 and Th2 only. Okay, Th1, Th2, I said the 3 main functions are differentiation into Th1, Th2, clonal expansion, and then migration to the area of infection. That's what that says. Th1, so let's talk about Th1 first. Th1, migration to peripheral tissues. Then you have the second antigen presenting cell. That's a macrophage. Macrophage, also phagocytosed, exactly the same pathogen, expressing exactly the same antigen on HLA2. If it counters that anywhere in the site of infection, you then get a fully activated effector Th1. Okay, so what I like to say is this is called a primed T cell. Primed means it's experienced the dendritic cell, so it's become primed. Now it's a fully activated effector Th1. That's a technical term. It's an effector Th1 cell. Okay? That's after it encounters a macrophage. Then you get release of lots and lots of chemicals in order to activate the macrophage. Okay? There's 2 that you need to remember. Interferon gamma, that is the main macrophage activating factor. If you don't remember any of the other ones, that's the one you should remember. Interferon gamma and tumor necrosis factor alpha, I think. TNF alpha. Yep, right, I am. Good. TNF alpha. So, INF gamma and TNF alpha. So that's here and here. There are other ones, as you can see, but not for you to know. Activate the macrophage. Yes? Now, the activated macrophage, I've said it becomes more potent, and the way it does that is it releases loads and loads of chemicals to kill stuff. Or it differentiates into one of those giant cells, epithelioid cells. So in this case, what I've said that it releases these sort of things to kill stuff. The main one here is tumor necrosis factor. It will kill stuff. tumor necrosis factor causes necrosis. It will kill stuff, a lot of stuff. Okay. Talking about Th1, he talked about granulomas a little bit. Okay. What a granuloma is the best example to think of is TB. What happens in TB is you have a bacteria called a Mycobacterium tuberculosis, and it's an intracellular pathogen. What cell does it infect? Macrophages, right? So what you have is this TB inside the macrophage. When it's not activated, it can't do anything about it, so it's walking around with TB inside it. Okay? Then it seeks help from a CD4 Th1 cell. It gets activated. Now it has the ability to try and fight the TB inside it. Okay? What it does that is it releases loads and loads of TNF, loads and loads and loads of tumor necrosis factor. So what you have is, this is what a granuloma looks like, and you should be able to draw, because it's in one of his slides, you should be able to draw this. You have a central area of caseating necrosis. Caseating, as he said, means cheese-like. That's a definition. Okay, it looks like cottage cheese. It actually does. It looks nasty. Okay, so you have, in this area, you have macrophages with a TB inside, and they're all releasing TNF. Okay, so this area is caseating. The cheese-likeness is because of the tumor necrosis factor killing not only the macrophage that's infected, it's also destroying the surrounding area. So it all becomes nasty.
So wait, what's the outline of your diagram? Is that
Okay, don't draw this yet. Don't draw it yet. This is me destroying loads of macrophages infected with TB, releasing TNF to try and kill themselves. Mycobacterium tuberculosis is really special, right? It's got this ability to withstand destruction. You'll do this in your lectures that you have this week, okay? Just remember, TB is resilient. It can't be destroyed easily. Yeah. So what's happening is the TNF is being released, but the TNF isn't really doing anything. It's not killing the TB. Okay. So in this area, you have all the neighboring, the good cells. Let's say this is the lung, right? All the good neighboring cells, they're all destroyed. So this is the central area of necrosis, but the TB remains in here. It's still not dead. It hasn't died. Okay. In order for it to do this, the first cell around, what do you think this is? This is the caseating granuloma. You have a layer of cells around it. What cells do you think these are? Based on what I've drawn here. Yeah, so granuloma, this is what you have to be able to draw in an exam. Label this caseating necrosis, central area of caseating necrosis. The first layer of cells around it should be activated macrophages. So these are activated macrophages. This is a giant cell, and the way you draw it is multiple nuclei. This is an epithelioid cell, so it's elongated rod-shaped. That's the first layer of cells around the central area of necrosis. Okay? What do you think is in front of this? What activates a macrophage? T cells. So you have lymphocytes outside. Does this make sense to people? These are lymphocytes. That's a granuloma. What's the point of a granuloma?
To wall off the infection.
Precisely. If this is the lung, this is a lung parenchyma, right? This is to wall off that area of TB. So it's a little spot. You'll see it if you look at a person's, like if you go to Whitechapel and you x-ray anyone, then you'll see this on the pigeons in Whitechapel have TB. I shit you not, it's true. Okay. Okay. If you X-ray a person's lungs, you'll see this. You'll see this central area. It's a little white dot. What happens in TB is over time, this granuloma's contained it to this area, but because you're getting necrosis, it's destroying the actual lung tissue. Right? So eventually, the TB will move out and then spread around the body. Okay? You'll see it on an X-ray. Why would you see this on an X-ray? What do you see on X-rays? Yeah, why is something condensed? Why is something white on an X-ray? Because it's got calcium. Okay? So over time, this becomes calcified, and you'll see little dots on the X-ray where this is. Okay? He's got a slide there with an X-ray on. Let's have a look at it.
That's a really different question. So how do they treat TB then
How do they treat TB? Well, antibiotics. Okay, TB is one of the hardest things to treat. Yeah, the TB's got resistant to most. There are special types of TB that are resistant, especially in Whitechapel, to most antibiotics. So you have your first line treatments of like rifampicin, pyrazinamide, all of that. I don't know if you've heard of those. You'll do it next week anyway. So that's it. Antibiotics, the only thing that kill it. Okay. Right. Nearly finished.
Was it, sorry, with TB, can you, how you say like it's all everything much, can you like walk around with TB and be absolutely fine?
Okay. In this audience, there's about, I don't know, 50 people or so, 20, 30 people. Okay. About 95% of you have been infected with TB. Okay. Probably all of you from Whitechapel. But what happens is you get primary TB, and then it goes into a latent period. And most people never reactivates. When you get immunocompromised, you get old, it will come back. And if you're genetically predisposed to be more susceptible, it will come back. That's what it is. Everyone's been infected. The BCG vaccine is bollocks. It doesn't really work. So yeah, everyone has TB in your mouth or somewhere. Okay. Are all granulomas caseating? No. You have non-caseating granulomas as well. So, okay, things that cause a non-caseating granuloma, things like talc, silicon, stuff like that. So the best example that I always give is talcum powder can cause a granuloma which is non-caseating. Don't ask me why. It just causes a non-caseating granuloma. The best place is in the ovaries. You get ovarian cancer. So women that powder their vaginas, right? Very often, it's not a good idea. You will get granulomas in your ovaries, and you will get ovarian cancer. So don't do it. And also, as I said, don't perfume your vagina. Don't do anything there. Don't perfume it. Don't roll around in mud. Don't do anything. Right? Because a lot of shit can happen. That's Th1. Does everyone understand that? That is activation of macrophages, all of that. Th2, very quickly, because I did it last week. Th2 I've done last week. Guys, there's only about 10 minutes left, right? So Th2, you have IL4, which converts T0 into Th2. Okay, migrates again to peripheral tissues. If it encounters a B cell, the B cell will get activated. B cell activation, what does B cell activation cause? Plasma cells, which release antibodies. So the B cell becomes differentiated into plasma cells. The main ones that you have to remember is IL-4, IL-5. Okay, there's also IL-3 and other ones. The main one is IL-4, IL-5. Okay, IL-13 as well, if you're really keen. Okay, what does IL-4 and IL-5 do that I talked about last week? What's the main thing? It releases the antibodies, yeah, fine, but a plasma cell does that when it becomes differentiated, with or without the help of the T cell. What does a T cell do to antibodies? Class switching. Remember class switching? Right? This is the one that's usually on a non-activated B cell. So you have IgM on the cell surface of a naive B cell. Okay? You get class switching into IgG, which is because it's the best antibody, or into IgE if you want to fight a helminth, because IgE is related to mast cells and to eosinophils, as I said. Okay? IgG doesn't really do anything, so there's never any class switching to that. IgA it's a bit different. Do you remember I told you? Okay, you know he hasn't actually officially taught you any of this, but he's mentioned a lot in the lectures about where you find IgG, IgA, and all of that. So what you need to know for antibodies, if you don't already know, IgG passed placentally from mother to child. Okay, it's the best antibody. I'm just write that in your exam. It's the best. Okay.
What is like the sorry, I know you told us earlier, but the E activates the eosinophils and mast cells, which is
IgG. Okay. IgG has three functions. It blocks pathogen binding, that's called neutralization, and agglutination. It activates complement. And it directly opsonizes, activates complement. So you know that classical pathway I said? IgG and IgM both do that, they activate complement. IgG also directly opsonizes. Okay? So that's IgG. The one thing that you want to know about IgG is it's passed transplacentally, yeah. Opsonizes stuff. So neutralization, opsonization, and complement.
What do you mean by neutralization?
Neutralization means, you know, all of these intracellular bacteria I was talking about, they have to get inside cells. Neutralization means these antibodies will bind to the bacteria and stop them from getting inside cells. So it blocks pathogens from binding. That's how I like to talk about it.
Is neutralization and agglutination the same thing?
No, neutralization is slightly different. Neutralization is the actual process of stopping it from getting in. Agglutination is the process of how it does it. Well, it's one of the processes by which it does it, and that is by binding to the pathogen. So if you have a pathogen...
Neutralization, like a way
No,
of
I'll draw this. If this is a pathogen, okay, agglutination means... Oh, shit. Yeah, the antibodies bind to it, right? So it's more difficult to move around. It's harder for it to infect cells. The process of it actually stopping it from infecting a cell is called neutralization, okay? And by this process, by the agglutination process, you're right, it can result in opsonization. So agglutination is quite central to all of it. But agglutination has its own thing as well that it does. It slows down the pathogen, it does lots of other things as well. Does that make sense in a simplified way? Okay? Right, so IgG, remember, transplacental. Passes placentally from mother to child, which is good for the child. Unless you have HIV, in which case it's bad. Okay. IgM is the main antibody on naive B cells. IgM is the main antibody on naive B cells, and it's a pentameric molecule. It exists bound to a J ring. Okay? So it's like, it exists as 5 IgMs bound to something called a J ring. This is how they ask you in EMQs. This antibody is found on naive B cells. What is it? And then you pick out IgM. Okay? IgA, the thing you want to know about it is it's found in secretions. So it's found in breast milk, it's found in your tears, it's found in pretty much any secretion, any bodily secretion. So gastric juices and stuff like that, they contain IgA. Okay, does that make sense? IgE, main thing is activation of mast cells, but it also opsonizes stuff for eosinophils. Just remember mast cells if you can't remember anything else. And IgD is a bit crap. What it is in the naive B cell, an early stage in development, you get expression of IgM and IgD, and then the IgD just dies off. Okay? Remember them in that order.
Can you repeat IgE?
IgE, which one?
For eosinophils?
Yeah, it opsonizes antigen for eosinophils. Yeah.
Can you repeat IgA?
IgA, okay, you find it in bodily secretions. So, for example, in your tears, in breast milk, stuff like that. And also, another thing is it's divalent, so it exists as a dimer, like this. So it's two of them joined together. Yeah, okay? Is that clear with everyone? That's all the B cell stuff done. Does everyone understand the Th2 response? Okay, lastly, I want to talk about autoimmunity. Okay, before I do that, I know you had a lecture on cytokines. It's a bit pointless because it should be covered as you go along. What's the definition of a cytokine? What is it? What is a cytokine? What sort of molecule? It's a what?
Glycoprotein.
A plasma protein. Okay. Oh, a glycoprotein. Yeah. It's a glycoprotein or a peptide. You can have its proteins, glycoproteins, whatever you want. Okay. What is the point of a cytokine? Yeah, so there's two ways that cells can communicate with each other. Through actually like binding to another cell via receptors, or via the release of cytokines which then bind to a receptor. So cytokines have to bind to a receptor for it to be recognized. Does that make sense? Okay,